Here we go. We're going to get started. It's 2 o'clock. Welcome to 2024, everyone. My name is Beth Foss. I'm the Director of Operations for the CRF, and we are starting a new series this year. And we're leading it off with a very new venture, new research that we're going to learn about. Dr. Troyk is with us from the University of Illinois Institute of Technology, and he's going to share with us, as I said, about new aids or visual prosthetics. We'll learn about more about that. For people who are at the end stages of choroideremia, people who might not be you or me, but that could be somebody in your family that you could give this information to. So it is really valuable, and we're going to take questions along the way if there are folks here that would like to ask them. But we also want to thank our sponsor, 4DMT, who helps promote these and support us in these endeavors. So we'll go ahead and get started. How are you today? Is Chicago warming up?
Yes, it is. Yeah. They're predicting 49 degrees next week. So,
Hey, there's some progress. What was the coldest that you got last week?
I think it was -10, something like that. Yeah.
And it
Yeah.
doesn't
It
mention.
happens now and then, doesn't last very long.
Well, we're sure great. We're really appreciative that you're joining us today. So thank you very, very much. I'm going to minimize myself. I'll staff the Q&A and chat feature, and I'll ask if anybody has questions.
Thanks a lot, Beth. And I'm very grateful to be here today and to tell you about our pioneering and innovative work in brain-based vision prostheses. I will be using some visual material, but I will make the presentation content completely audio so that those who may have visual issues won't have to rely upon the the visuals to see, but they'll be there when this is available on the website afterwards. So let me just introduce myself. I am Philip Troyk. I am a professor of biomedical engineering and the executive director of the Pritzker Institute of Biomedical Science and Engineering at the Illinois Institute of Technology in Chicago. And I am going to tell you today about a project that we have been working on in cooperation with several institutions, including quite prominently the Chicago Lighthouse for people with visual impairment or blindness. So the name of this research is the Intracortical Visual Prosthesis, or ICVP for short. And you may wonder, well, what does that actually mean? So intracortical means that it actually penetrates into the cortex, which is part of the brain. It is an FDA-approved clinical trial that is funded by the NIH Brain Initiative. And this is basically a brain interface system in order to explore how artificial vision might be provided and be helpful for people who have profound blindness. So technologically, it's like a miniature cell phone network in the brain. So it's comprised of miniature modules that are 5 millimeters in diameter and less than a millimeter thick. And for perspective, that's about the size of the little button that's on the positive end of an AA battery, a little thinner than that. So these tiny little round modules contain electronics. They contain very fine wire penetrating electrodes, and they contain a receiving, transmitting antenna. And each one of them is literally like a region for a little cell phone network in the brain. The electrodes penetrate into the brain. The little device sits on top of the brain. And the electrodes penetrate about 10 thicknesses of a piece of paper, about 1 millimeter or so down into the brain. And there's 16 of them on each of these little modules. These little modules are called wireless floating microelectrode arrays, or WFMA for short. Every one of these little modules has an identifier that you could think of as an area code. And every electrode that's within these modules, the 16 electrodes that's within each module, each electrode has an identifier that you could think of as a phone number. So by transmitting information into this little network that is residing on the top of the brain, you can dial up an area code, you can dial up an electrode, and you can tell it what to do in terms of injecting tiny electric currents into the brain. So the idea is that these little currents injected into the brain in the area that is normally receiving information via the optic nerve from the eyes. This is called the primary vision cortex of the brain, that if we stimulate with these tiny electrodes in this area, that then the brain perceives as though it was receiving visual input via the optic nerve from the eyes and the retina. So it's a perception. We're not actually putting light into the brain. In fact, a person who would have this doesn't even need to have physical eyes. They could have eyes that are enucleated. They could have a damaged optic nerve. And we're bypassing the optic nerve and the eyes, and we're going directly to this area of the brain, which is on the side of the head, kind of where your skull bends down towards your neck. If you put your hand up, you put your palm up there and you feel it. On each side of the brain, that's your occipital lobe, and that contains this primary vision area that we're making this little miniature cell phone network interface to. And the idea is that you capture an image with a camera, you translate it into signals that then are sent to these tiny electrodes that are in these little modules. And the idea is to create the perception in the brain that corresponds to the image that's captured by the camera. These electrodes are so tiny that if you compare them to a human hair, you could put about 20 of them on the tip of a human hair, because they're extremely tiny and highly engineered. Now, this project has a long history. Our part of the project actually began in 2000, but it was an outgrowth of a much earlier project that was started at the National Institutes of Health in the 1970s. And from the 1970s to 2000, the National Institutes of Health funded a lot of research to develop basic technology that could eventually come to the point where we are today. In 2000, we assumed the leadership of the project, and that led up to an FDA approval in 2019. And in 2022, the first blind participant was implanted. So the project has a very long history and a lot of research and a lot of people who have dedicated substantial parts of their career to this work. So think for a minute how vision works. Light normally enters the eye. The retina converts that light to neural signals. The optic nerve carries those neural signals to the brain. The brain decodes those neural signals, and the brain creates the perception of image using a very complex neural machine, which we call the brain. And about 1/2 of the human brain is involved with some form of vision perception processing. So that's a lot of neurons, a lot of neural machinery And the idea here is to bypass the eyes and the optic nerve and put artificial signals directly into the brain at the location that normally receives the signals from the optic nerve, bypassing the eyes and the optic nerve, and put artificial vision information directly into the brain. So the idea is that with these little miniature modules, each one of them that we can talk to wirelessly with no wires that cross the scalp, no connector on the head, as you may have seen for other systems, these wireless, fully implantable modules that are about the size of the button on an AA battery, a group of them are implanted into the brain under the skull. And by communicating with them and talking to them, we're able then to send artificial vision information directly to the brain for the purpose of trying to create the normal perception of vision in the brain, but bypassing the eyes and the optic nerve. Now, it's important to have expectations about such a system. This is at the very early pioneering stages of this technology. It's not meant to restore natural biological vision. It's not meant to have someone see their loved one's faces or to replace the function of a cane or a guide dog. It's not intended for reading. It's not intended to watch television. We just don't have that type of visual resolution with this early stage technology. But it could provide a very crude form of artificial vision. Imagine if you hold up a kitchen colander that is used to strain noodles that has holes in it, and you hold it up in front of your face and you move it around and you look through those holes to see what the world around you looks like. That's somewhat like what this crude artificial vision is like. So I'd like to pause here and ask if there are any questions about the basic technology, the basic concept. I'm going to be going more deeply into the technology. I'm going to be going into what the qualifications are to participate in the study and what the early results are that we have from a participant who's implanted. But if there's any questions about vision, about the basic concept of this, I'd be happy to take those questions now.
You know what? We don't have any. We've got a couple people that joined us, but nobody has any questions yet. So you're good.
You know, when I teach my classes, if I don't have questions, it's one of two things. Either I've been exquisitely clear in how I've explained it, or everybody's bored to death. So I'm going to pretend that it's the first part. And as we go on, there may be additional questions and we can take them at that time.
You know what? Let me ask you this. We did have somebody come in and ask, how invasive is the procedure?
Yes, so I'm going to explain that, but I will tell you that it involves what is called a craniotomy, in which a neurosurgeon will open the scalp, will take out a section of the skull that's about the size of the bottom of a soft drink can, will then put the devices in, replace the skull, close the scalp. And for a neurosurgeon, this is the easiest thing they can possibly do. It may sound highly invasive, but they do it every day, and it's a very easy procedure to do, and it's relatively low risk. So it does involve brain surgery, but there's actually no penetration into the brain because the little devices are just kind of popped in, and the little tiny electrodes go into the brain and they sit on top. So the whole procedure, as I'm going to explain, can go very quickly.
All right. Well, I'll let you take it away then.
Thank
Share
you.
more on that note.
So I'm going to tell you a little bit more about the technology. And so I said these wireless floating microelectrode arrays are miniature electronic devices that contain an electronic chip, just like the chips that are in your mobile devices, except we designed this one ourselves exclusively for this. It contains a receiving antenna, which is like a little coil that couples with a transmitting device that's outside on the head, and it contains 16 miniature stimulating electrodes that then penetrate into the brain. If you look at it under a microscope, it looks a little bit like a little miniature hairbrush with the little needle electrodes sticking down and the little flat top. It's like a thumbtack that has very tiny little electrode tacks sticking out. It's about 5 millimeters in diameter and less than a millimeter thick, which I said is about the size of a button on an AA battery. The antenna coil that's inside receives power from outside of the scalp from a transmitter device, which looks like kind of like a donut that goes on the back of the head. It receives power into the device, and then we can talk to the device. It's very similar to the near field charging that we have for our mobile devices, our phones, our pads, where you just place it on top of a charging device and you don't have to have any physical contact. There are no wires that penetrate out from the head. The scalp is completely closed, and after the surgery, there's no visual indication that anything is implanted. The devices are meant to be implanted chronically, and they are far less invasive than many, many other things that are implanted routinely into the heads of people. Now, the electrodes are very, very tiny, and your human hair, if you looked at the edge of it under a microscope, it could hold about 20 of these electrodes standing on end on the tip of the human hair. So they're very, very small, and they're made to penetrate into the brain about a millimeter, which is about the thickness of 10, 15 pieces of paper. And those little electrodes reach the neurons that are in the brain. They put tiny electric currents, they stimulate the neurons, and then the neurons start to process signals similar to what they would get when they receive those signals from the optic nerve. So in February of 2022, in fact, on Valentine's Day of 2022, our first recipient received 25 of the WFMA devices comprising 400 electrodes implanted on the right side of their brain. This individual has no light perception. The surgery was done at Rush University Medical Center in Chicago by the chairman of neurosurgery, and it was put on what's called the dorsolateral surface, which just means kind of the top surface of the occipital lobe. And the occipital lobe is located on each side of the head, right as the skull starts to bend down towards the neck. That's where your occipital region is. They're actually put in using a little inserter tool device that kind of shoots them into the brain very quickly. And the top part of the thumbtack sits on top of the brain, and the tiny electrodes penetrate about a millimeter into the brain. The total surgery took 4 hours, and most of that time was spent in setting up, doing the craniotomy, doing the closing, because when we implanted the 25 WFMA devices, we were implanting them at about 1 a minute. So it goes very quickly, and it's very easy for the neurosurgeon to do. After the surgery, our participant returned home, I think after a day, and then recovered for 1 month. There were no post-surgical complications, no medical issues. And in the past 22 months, we have tested the implanted technology, which shows very high electrical stability. We have tested the amount of current that we have to put into each electrode in order to get a perception. We have done what's called mapping, which I'll explain more about in a moment. We've hooked up a camera, and we've looked at what can be done for visual task training, testing, and functionality. We had planned to implant about 192 electrodes in a smaller number of devices, but with the surgeon's discretion, we were pleased that he decided to implant 25 WFMAs to make the 400 total electrodes. I'd like to stop and ask if there's any questions before we might talk about the functionality that one can achieve that we've seen with such a system. Are there any questions about the technology or the surgery that is involved?
We do have one. Jim asks, how much vision does a participant need to have?
So our clinical trial has what are called inclusion and exclusion criteria. That means the conditions for a person to participate. And right now, as we agree with the FDA, the protocol is set up for participants must have no light perception or bare light perception. What that means is they have to not be able to see a hand waving in front of their face, or they have to not be able to determine where a lamp would be on in a room. They can notice that a lamp is on or off, but they can't have vision that is sufficient for them to localize exactly where that lamp is. We give them some sophisticated vision screening tests in order to assure that the vision is at an adequately low level. And the reason for this, which we agree with, is that the FDA did not want us in this pioneering study in which there are unknowns to compromise any residual vision that a person may have now. So we carefully screen to make sure that there is no or bare light perception. Any other questions about the technology or the surgery?
No, not right now. You're doing great. Thank you.
Okay, then I'm going to continue and I'm going to tell you about some of the functionality. So when we put these electrodes in the brain and when we start to stimulate them with the tiny electric currents, the recipient, the participant, perceives what seems like little spots of light in front of them. It's not real light because we're putting it in artificially, but the brain thinks it's light because we're stimulating the vision part of the brain. And these are called phosphenes. People can experience phosphenes if they wake up in the morning with their eyes closed and kind of rub their eyes and you see little spots. That's similar to what phosphenes look like. Because we're stimulating the vision part of the brain that is very sophisticated and normally receives the input from the optic nerve from the retina, stimulating artificially produces a variety of different vision perceptions, all of which are called phosphenes, but they can have different shapes, different colors. Some may move, some may have texture, some may look kind of shimmering. It depends upon where the electrodes are, but we can't know where in the vision field the person would perceive these phosphenes. So when we stimulate an electrode, we have to ask the participant, where do you see these? Where do you see something? And they point in the center of them, and they point off to where they see something for each of the electrodes that we stimulate. We have 400 electrodes. So you might imagine it takes a little while to do this, and we have to do it more than once so we can gain confidence. This is called mapping. We're looking at what is seen and where is it seen. And then we create a catalog of what can these electrodes produce, what are the phosphines, where are they, and we know for each electrode when we call it up, it will produce a spot in a certain portion of the vision field of a certain character. So far, we've been able to map the location of about 20 of the 25 devices. Not all devices end up being in a portion of the brain that gives phosphines. It could be in a portion of the brain that processes those phosphines somehow. So out of the 25, we have a very good response for 20 of them, and we have mapped where they are in the vision field of our participant, which is in the lower left portion of their vision field because we've implanted on the right side of the brain. And we now have a catalog of the phosphines that are produced corresponding to each of the electrodes. This is called the map. When we did the implantation in 2022, by August, we had maybe 4 or 5 locations that we could determine where phosphines were. By March of 2023, we had a much larger number. And by September of 2023, we had more of them and understanding more of their shapes. Some are tiny pinpoints. Some are the size of a nickel at arm's length. Some are brighter, that represents a larger area. Some are dark. Some are elongated, like little ovals or little sausage-like shapes. And as I'm sure many of you know, many people with blindness don't have darkness. They have sometimes visual noise that is kind of like the static on a television. And so when we stimulate some of these, sometimes it makes it dark and it quiets that noise in a little location. So we now have a pretty sophisticated map of a collection of these that then we can use as the building blocks of the artificial vision. Now, natural vision compared to artificial vision needs some context. In your eye, there's about 125 million photoreceptors. In the brain, where we're putting these implants, there's about 350 million cortical neurons. We've implanted 400 electrodes. So you can imagine that any artificial vision that could be produced by this entry-level system will be very crude compared to natural vision. We are not at the point where we can put in the science fiction kinds of things that you'll see in movies and television shows in which there's little miniature robots crawling around communicating with your brain cells. We are at a point of where we can make the interface with 400 as opposed to 350 million that are there biologically. And yet we're looking to see how this artificial vision might produce some beneficial sensory input. The system is not meant to restore natural vision. It's not meant to see loved ones' faces, replace a cane or a guide dog. We can't expect that people can read or watch television with it. But there are some surprising things that it can help do based upon what we've seen in our first participant. It can help to determine where large objects are in the environment. With an infrared camera, it can easily locate people in a room. It can help determine the location of high contrast walkways or the location of door frames. Our participant can find high contrast objects on a table. A person could determine whether the lights in a room are on or off or where windows are, possibly crosswalk lines, possibly large moving objects like cars. So if you think about normal vision and all of its eloquence, and you think that, okay, maybe it'll be degraded and it'll be pixeled a little bit. In reality, if you think of the kitchen colander analogy, it'll be much cruder than that. And we often say that basically we are trying to provide someone who is totally blind with artificial vision that would make them like someone who is legally blind. So it's important to have the expectations of what can be done at this stage of the technology. But it is pioneering technology, and it has now accomplished something that has never been accomplished before, which is indeed this fully wireless, implanted, networked system that is interfacing with the vision area of the brain. I'd like to stop and ask if there's any questions before I go on and tell you about some of the vision tasks that our participant now has been trained with.
Well, here's some questions, and if you want to answer them later on, that's okay too.
I'm happy to answer them now.
Okay. Do you have any thoughts on how this device compares to others currently being developed?
Well, yes, I do. The others that are currently being developed that go into the cortex the way ours do not have full wireless capability. The predominant one has a connector that goes on the side of the head, and it's only approved for about 6 months of being implanted. There was another system that was tested by a company called Second Sight, which also made an Argus retinal prosthesis system. They tested a system called Orion, in which much larger electrodes sit on the surface of the brain rather than penetrating into the brain. That system was tried in 6 people, and that system is no longer available for use. There are other approaches worldwide that are being looked at, but the ICVP, the Intracortical Visual Prosthesis system, which we are now testing in the clinical trial, is the only one that has full wireless networked capability that has penetrating electrodes that go directly into the brain. And if any of you have heard about the stuff that Elon Musk has talked about, and if you happen to know him, you can tell him we've already done what he's trying to do. So this system is indeed the most sophisticated, and it is meant for chronic implantation. There is no need to have explantation of the system. Are there other questions?
Are you working with other organizations or researchers, device companies that would be using your technology as like a platform or the foundation to enhance visual acuity moving forward?
That's a very good question. Our project involves 8 institutions, and I'll describe all those at the end, but it does include 2 companies. And yes, of course, the goal is to take what we learned from this study and use it as a technology platform to understand how we can sophisticate the technology moving forward. Indeed, we know that all of our electronic devices, computers, tablets, phones, they all sophisticate as time goes on. The inaugural devices have encouraging capability, but it does sophisticate as we learn more about how to use them and technologically develop them.
Good answer. How many people have been treated so far with this device?
Presently, we have 1 participant, and the clinical trial that we have allows for 5 total participants. So we are now recruiting 4 additional participants.
Hey, here's one more question. How do images that the eye would see get naturally communicated to the electrodes in the brain? Right now, are you just pushing a signal to the brain?
So to replace the function of the eye, we use a video camera. In our present system that we're testing, the video camera is very small. It's mounted in glasses that the person wears, and the camera sits in the bridge of the glasses between the two lenses over the nose. That camera captures the image, and then it goes through a computer image processor that decodes the image and then converts it into the communication signals that we use to go to each one of the electrodes. And it tells which electrode to come on at which point in the vision field in order to correspond to where the image is. Kind of like pixels on a computer screen.
That's all the questions we have at this point.
Okay, then I'm going to proceed and I'm going to tell you something about the functionality that our current participant has experienced. So in the past 22 months, we have done this mapping and we've carefully looked at what perceptions occur, including the dots that we call phosphenes that are light and dark. But in some cases, they look like lines or shapes, diamonds, squares, moving lines, shimmering shapes, or shapes with colors. We've connected this mapped system through the wireless interface to the camera that captures the image that is contained in the glasses. And our participant is able to do some surprising visual tasks with them. So what I'm showing that you can't see right now, but I will describe verbally, is our participant is seated in front of a table with a black tablecloth. There are 4 objects on the table: a cup, a bowl, a spoon, and a plate. And in this type of test, we ask the participant to scan the table as the participant moves their head back and forth, looking at these little phosphenes as they illuminate on and off, and determine where I'm sorry, I'm trying to get back to turn on the video, where the object is that we've asked them to pick up. So the 4 objects, the cup, the bowl, the spoon, or the plate, are randomly placed in an order about maybe a foot apart in front of the person seated at the table. And then we randomly choose one and we say, okay, find the plate and reach out and pick it up. And so our participant will scan around the table, looking at the shape and the size of the objects by seeing which of the phosphenes, or as you might imagine with the colander, how many of the holes seem to be bright, and then can reach out and pick up the object. Our participant presently gets it correct about 70% of the time. By just guessing, as you might imagine, it's less than 2 out of 10 times they can get something correct. As another demonstration of what could be done, our participant themselves said, you know, I often spill pills. And I think I could use this system to find a pill. So we said, that sounds a bit challenging, but go ahead and try. And so we did try it, and we did some trials. And by putting what is a white Excedrin pill on a black tablecloth, albeit with high contrast, by scanning around some, our participant is able to find the pill, localize where it is, and reach out with the hand and pick it up. So what else is the person able to do? Well, our participant right now can locate people in a room. We recently had a test session in a larger room in which there were about 11 or 12 people in the room, and our participant could scan around the room and could point to where the people are in the room. Our participant can determine the location of a high contrast walkway that's on the floor. Our participant can determine where people are in a corridor as they're walking with their cane. They do that using an infrared camera that picks up the signature of people because of their heat. They can also identify with scanning single letters projected on a large screen in front of them, or determine the direction of alternating white and dark bars. We are now investigating how this could help for mobility enhancement. And we are learning every day as we start using the system with the camera. We've been using the system with the camera for about 1 year. And during that time, our participant has trained, has learned what the perceptions are that they're getting, has learned strategies for being able to utilize such a system, and most importantly, is helping us to understand the capabilities of such a system so that we can do further development in the future. We think of the participants in our study not as human subjects. They're certainly not patients. They're not sick. They are volunteers who are giving of themselves to help us explore and go to a place that no one has ever been to, which is how do we learn the language of talking to the visual area of the brain with the artificial vision so that one day something like Geordi's visor on Star Trek can become a clinical of care reality. In this regard, we think of our participants more as analogous to astronauts who trained, developed capabilities, tested technology for being able to go to a place that no one had been before and tell everyone else what was there. Our subject is able, our participant is able to tell us what is experienced, what is perceived, and how the stimulation produces different effects. Now, we are recruiting additional participants. We're recruiting 4 additional participants. And the basic requirements are that participants must have no light perception in both eyes. I explained that earlier. That means no or bare light perception in both eyes, so that they cannot see a waving hand in front of their face. They must have had correctable vision up to the age of 10. The purpose of this is because unless a person has had normal vision in the early stages of their life, their brain doesn't develop in the same way. The vision area will develop differently. In the future, it is quite likely that such systems would be used even for people who are born, children who are born without vision, just as cochlear implants started out being used for people who were profoundly deaf and now are implanted in children. Our participants must have had blindness for at least 1 year. And that's because this is not about vision restoration. It's about gaining knowledge about how an artificial vision system could work. And we really need our participants to be well adjusted to having blindness, and they must have had some form of blindness rehabilitation, either cane training, guide dog training, some form of blindness rehabilitation, again, so that they are adjusted to having blindness and they're not looking at this as a way to try to get their natural biological vision back. They must be between the ages of 18 to 71. So the cutoff age is 70. And the reason for this is because past the age of 70, there are some increased risks involved in the cranial surgery. And most importantly, the motivation should be research and exploration, not vision restoration. Our current participant is extraordinarily good in this regard. The person understands very well what this is and is fully engaged with us and often helps to make very valuable suggestions about how to change the testing or enhance the information we're getting. So if any of you listening may yourself or know of someone who you feel might be a candidate, we have a screening process that takes place. And if you want more information, there are two ways that you can get that. One is to send us an email at our email contact, which is icvp@iit.edu. That's icvp@iit.edu. This is a dedicated email that if you send inquiries to that email, someone will contact you back, usually within a day. You can get much more information, including prior webinars, newscasts that have gone on about this project at the Chicago Lighthouse website. And that website is Chicago Lighthouse, all one word, .org/ICVP. That's chicagolighthouse.org/ICVP. You'll find a lot of information about this project there, including prior webinars and newscasts and articles. The team is made of people from 8 institutions, very dedicated, which includes my university, the Illinois Institute of Technology, Rush University Medical Center, the Chicago Lighthouse for the Blind, Johns Hopkins University, the University of Texas at Dallas, Microprobes for Life Science, a company in Maryland who makes the electrodes, Cygenics Incorporated, which is a company in Chicago that makes the electronics for this, and the University of Chicago contributing vision science and research analysis. So I've been very happy to tell you about our project today, and I'm happy to take any follow-up questions that you may have. You're muted, Beth.
Oh, there we go. Thank you. You know, it is super exciting, and it just provides, as I was actually texting with Kathy here, provides a lot of hope for people. And I very much enjoyed your analogy of the astronauts. I think it's exciting, and it provides encouragement and hope, hopefully for the future. We do have a couple more questions that came in. First of all, are you acquainted with Dr. John Pizarris, I believe is how you pronounce it, at Mass General? It seems there might be a potential for collaboration. And then there's a link here in the questions.
Yes, I know John. John has been working at trying to do an electrical vision interface at a portion of the brain that's called the thalamus. What happens is that the optic nerve starts in the eye and it stops off at this region of the brain called the thalamus, which is like kind of a sensory switching station. And then from there, it goes to the vision cortex where we are. So John has done a lot of work looking at trying to do the same type of electrical interface at the thalamus. There have been research projects at the retina, at the optic nerve, at the thalamus, and at the brain. Our approach is to be in the brain.
Next question. Do participants have to live in the Chicago metro area? How often are follow-up visits?
That's a very good question. When we started this study, we had hopes of really recruiting everyone from the Northern Illinois area. That actually didn't work out, but of course, our study started in 2020, And in the middle of 2020, a lot of people had a lot of other things on their mind other than going to a hospital to get elective brain surgery. So we have expanded the recruitment to be really nationwide. And we do work with any interested candidates for the transportation and for the logistics. So from the time that a person would actually be accepted for surgery, which is prefaced by having a screening process that we go through, medical, psychological screening, other kinds of screening. But once surgery is done, then the person would have to be able to be available. It depends, but maybe once a week, twice a month, depending upon schedule. Our current participant started out, does not live in the Chicago area. And our current participant started out coming once a week for 2 days a week, but then went to twice a month, each for 2 days a week. We arrange for the logistics of transportation. Other candidates have suggested to us they'd rather come for 1 week and then not for the rest of the month. So we're very, very flexible on how that can work. After about a 6-month period, the testing sessions could drop off dramatically, particularly now because we've learned so much from our first participant after almost 2 years. So this person has remained fully engaged and has been coming in, and we appreciate that very much, that dedication. We realize that people have lives, they have jobs, they have family, and we work with them around that so that if someone is interested and is a good match, that we don't let the logistics of it really be a barrier to participation.
Here's another good question I was going to ask as well. Is there financial assistance available regarding transportation and housing?
Absolutely. There is no cost to any participant for participating in this study, but there's no remuneration to the person. So you don't get paid for being in the study. But we cover the costs of transportation, of housing, of meals while they're here. We don't want this to be a financial burden to anyone, so we do have funds available for that purpose.
Dr. Troyk, what do you think are the long range goals? So say you have 5 more, so you have your panel is 6 objective, that would be the goal. You would do the study for how long, and then at what point would you be done, and then what's the next step?
So the original study was designed to be a total 5-year study, of which the first 2 years was for us to get approval from the FDA. That we did. There was then the plan to have a 3-year clinical portion in which we would implant 1 person in the first year, 2 people at the beginning of the second year, and 2 people at the end of the second year. It didn't quite work out that way. And I'll blame COVID for a lot of it, okay? But so we are now in an extended period of the trial. And so generally, people who would be implanted now could figure that they may have 2 years, possibly a little longer of participation. When that study is over, we will then have the data from 5 people, and we will then have a pretty good idea of, at the level of technology that is technologically possible now, what type of vision function could actually result and how useful could it be. That would then guide a next step, which would be how could such technology become clinically available and what would be the criteria for being able to get it. So a lot of the future of this depends very much upon what we find out. But in 5 years, are we going to have totally natural vision restored? No, that's not going to happen, no matter what science fiction you may hear from what source. However, we've already been surprised. at some of the things that are useful. Our participant, in fact, has said to us, my dogs actually, I think their dogs are deaf and maybe almost blind now. And they said, sometimes I trip on them. I'd love to be able to tell where my dogs are. And it may seem like a small thing, but in fact, that would be pretty easy to do with this system.
That's awesome. That really is great. Are you able to tell us at all, roughly the age of this patient that's treated?
I'm not be able to get information specific about this. If you do look on the web, even on the website, there is information about the person that they have volunteered themselves.
Sure.
Okay, so per the study protocol, we're not able to give out personal information about the individual, but there is information out there that one can easily find. And so that's not too difficult, but I can tell you that they're less than 70 years old.
I don't have any other questions, and we don't have any more coming in either.
Okay, then I appreciate
Great.
very much having the opportunity to present to this group. If anyone here has any interest or might know of someone that has an interest, again, the email address is icvp@iit.edu, and you will get a prompt response from this.
And someone else just wrote in, very interesting and thank you for your presentation, which I'll echo. Thank you so much. Thanks for everything. And we just look forward to hearing what more information you'll have maybe next year. That'd be great.
Thank you very much for the opportunity to talk to you today.
You bet. Thanks everybody for joining us. Oops, wait 1 second. There's one more question.
One more question? Go ahead.
Nope, it was just another thank you.
Okay.
All right. Thanks, everybody. Thanks
Thank
again.
you.
Have a great rest of your week. Bye-bye.
Bye.